I want you to do something for me right now. If you aren't driving, obviously, just take a second and look at your hand. Like, really look at it.
Yeah, flex your fingers a bit.
Exactly. Curl your fingers into a fist and then open them back up. Now take a slow, deep breath in and let it out.
It feels pretty mundane, right?
Right. But here's the crazy part. Every single part of your body that just performed those actions, the muscle fibers contracting in your hand, the nerves firing that signal from your brain, the lung tissue expanding in your chest, all of it started as a single cell.
One single cell?
Yeah, a cell no bigger than like a full stop at the end of a sentence on a page. And that brings us to the mission of our deep dive today. We're trying to unpack this massive mystery. How does one microscopic speck of matter, with no architect, no supervisor, just know how to build a fully functional human?
It really is. I mean, it's one of biology's most extraordinary feats. We're looking at a system that basically assembles staggering complexity from absolute scratch.
And it does it so reliably.
Breathtakingly reliably. And to really understand how it pulls that off, we have to look at the exact starting point. So the story officially begins with a process called fertilization.
Right, the very beginning.
Exactly. This is the moment a single sperm cell and a single egg cell fuse together, and they create one entirely new cell, which in biology we call the zygote.
The zygote. It's completely wild to think that this one microscopic speck contains the absolute complete starting material for a whole person.
Oh, yeah. Everything is in there?
I mean, my brain, my fingernails, my heartbeat, it's all packed in there. But what is actually inside that zygote that's driving the whole operation?
Well, it all comes down to a famously long molecule you've definitely heard of, DNA.
Oh, of course.
Yeah, that single cell contains a full set of genetic instructions which you inherit half from each parent, and it's all wrapped up in DNA. Now, you will constantly hear people describe DNA as a blueprint for a human being.
Wait, blueprint of life, right. That's basically the marketing slogan for DNA. I feel like I saw that in every high school textbook.
Yeah. It's a very catchy slogan, but we really need to correct that misconception right away. It gives you a deeply misleading mental image.
Misleading how?
Well, think about what a blueprint is. It's a fixed static diagram. A builder looks at a blueprint, reads the whole thing to see where all the walls go, and follows it once to build a house.
Okay, yeah.
DNA just doesn't work like that at all. It is much more accurate to think of DNA as a massive encyclopedic recipe book or like a giant construction toolkit.
Okay, I can picture a giant recipe book.
Right. And inside this massive recipe book are thousands of individual instructions, which we call genes.
The recipes themselves.
Exactly. These genes are essentially recipes for building thousands of different proteins, and proteins are the tiny molecular machines, the bricks and the mortar, that actually do the physical work and build the structures inside your cells.
So if we run with the cookbook idea, if I have a massive encyclopedia of recipes in my kitchen, I don't walk in and cook every single recipe in the book all at once for one Tuesday night dinner.
No, that would be a disaster.
Right. I pick the pasta recipe, I completely ignore the cake recipe, and I just use what I need for that specific meal.
And if we stick with that kitchen idea, the secret isn't just having the recipes, it's knowing exactly when to cook them.
Timing?
Yes. Not every gene in that DNA recipe book gets used at once, and they absolutely aren't used the same way in every cell. The exquisite coordination of switching specific genes on and firmly switching other genes off at very specific moments is what actually drives this entire building process.
And there's a term for that, switching, right?
There is. We call this switching process gene expression.
Gene expression. Okay, so the zygote isn't just reading a blueprint from start to finish. It's constantly flipping through the cookbook, deciding which recipes to express and which to just ignore.
That switching on and off is the beating heart of development. But, before the zygote can really start specializing its recipes, it has to tackle an almost absurdly simple first step.
Which is?
It divides in two.
It just splits right down the middle.
It does. We call this cell division. The one cell becomes 2, then those 2 cells divide to make 4.
4 becomes 8.
8 becomes 16, and it just doubles repeatedly. But here is the critical part to remember. Every single time a cell divides, the new daughter cells receive a complete, identical copy of that original DNA recipe book.
Cover to cover.
Cover to cover.
Wow. That sounds like a massive logistical headache. I mean, we are talking about trillions of copies of an enormously long molecule.
It is, it's really worth marveling at the sheer reliability of this copying process. It happens over and over with remarkably few errors.
Which I guess is pretty important.
Vital, actually. because a serious mistake at this early stage would cascade into every cell that comes after it. So for those first several rounds of cell division, the cells are just multiplying. They don't look or behave like any particular body part yet. They are just building up the raw supply of building materials.
Okay, I have to jump in here because this brings up a massive logical hurdle for me.
Let's hear it.
If these cells are just blindly copying the exact same recipe book over and over, and every single cell is totally identical, wouldn't you just end up with a massive, shapeless blob of identical cells? Like, how do we ever get from a uniform blob to an eyeball or a heartbeat or a patch of skin if they all have the exact same manual?
That is the ultimate hurdle the embryo has to overcome. Because you're right, a shapeless ball of identical cells doesn't get you a functioning human.
Right.
At a certain point, these completely identical cells have to start becoming different from one another. Some are destined to become skin, others muscle, others nerves. This progressive process of becoming different is called differentiation.
Differentiation. Okay, so this is where the gene expression we talked about really kicks in. The cells start picking different recipes out of the identical books they hold.
They do. With very rare exceptions, every single cell in your body carries the exact same DNA recipe book that was in that original zygote.
Which is so counterintuitive to think about. Like, my skin and my nerves have the exact same instructions.
They do. They are astonishingly different in how they look and function, but the instructions inside them are identical. The difference is simply which pages of the book they are actively reading.
So how does a nerve cell use the book compared to a skin cell?
Well, think about what a nerve cell needs to do. It reads the genes required to build, long branching cables and electrical signaling machinery. But just as crucially, it firmly switches off the unrelated genes.
Like what?
For instance, a nerve cell has the genes for making tooth enamel sitting right there in its DNA. It has the genes for making stomach acid, but it keeps those pages firmly closed.
Oh, wow.
And the skin cell does the exact reverse. It turns on the skin-making genes and shuts down the nerve-making genes. Differentiation is this step-by-step narrowing of which genes are active until a cell finally commits to a specific permanent identity.
But wait, if I'm a cell sitting in this growing ball and I have the whole book, how do I know which page to read?
That's the big question.
Right. What tells one cell to turn into skin and tells another cell, just a fraction of a millimeter away, to turn into a nerve?
The answer involves some of the most elegant chemical choreography in all of biology. The embryo uses these chemical signaling molecules called morphogens.
Morphogens. Kind of sounds like morphing or changing shape.
Form creators, literally. A morphogen is a chemical signal released from a very specific source within the embryo, usually just a small cluster of cells.
Okay.
These source cells pump out this chemical, and it spreads outward, diffusing through the surrounding tissue, and this creates a concentration gradient.
Concentration gradient. Let's try to visualize that.
Yeah.
Like dropping a single drop of dark blue dye into a glass of still water.
That works perfectly. At the exact spot where the dye hits, the water is a deep, intense blue.
Right.
But as it slowly spreads outward, you get rings of fading intensity. A little further out, it's medium blue. Further out still, it's light blue until eventually the water is barely tinted at all.
So it's almost like a cell is listening to a radio broadcast.
I like that.
If the cell is bathed in a massive concentration of the chemical, the signal is blasting. And the cell says, okay, message received, I'll turn into skin.
Right.
But if the cell is further away and the signal is just a whisper, it says, got it, I'll become a nerve. The cells are basically just reading a chemical dial to figure out where they are.
They are reading a dial, and they are remarkably sensitive to it. A cell bathed in a high concentration switches on one developmental program. A cell experiencing a middling concentration switches on a different program entirely.
That is incredibly economical. You don't need a million different signals, you just need one signal at different volumes.
It is economical, and it's also highly collaborative. The morphogens establish these broad fading zones, but cells are also constantly talking directly to their immediate neighbors.
Oh, they talk to each other.
Yeah, they physically touch each other and use short-range signals to say, in effect, hey, I'm becoming skin, you should become skin too.
Oh, peer pressure.
Exactly. This neighbor-to-neighbor whispering takes those broad, faded zones and sharpens them into crisp, clear boundaries between different tissues.
And once a cell gets that initial message from the broadcast or the neighbor, does it just instantly transform?
No, it's a cascade. We call them gene cascades. One gene gets switched on by the morphogen, and it produces a protein whose only job is to go and switch on 5 other genes.
Oh, I see.
And then those 5 switch on 20 others. So a simple early instruction ripples outward into this elaborate pattern. A cell doesn't wake up and decide its fate in isolation. It becomes based entirely on where it's sitting in the embryo and what signals are washing over it in that exact location.
So these fading chemical signals are basically creating a three-dimensional map for the cells.
They create a literal coordinate system for the body. Very early on, one gradient defines an axis from head to tail. Another gradient, coming from a different direction, defines an axis from back to belly. Eventually, another sets up left to right. Every cell reads its exact location on the head-to-tail axis and the back-to-belly axis simultaneously.
Every cell basically has a biological GPS telling it its X, Y, and Z coordinates.
Yes, and that combined GPS data tells it exactly which pages of the recipe book to open. If this early coordinate system gets set up even slightly wrong, the errors cascade through everything built on top of it.
That brings up a really fascinating question about destiny, actually.
How so?
If a cell's entire future is tied to its location and its GPS coordinates, is it locked in immediately? Like, can a cell ever change its mind if it gets moved?
To answer that, we have to look at a vital concept called potency. Potency is a measure of how many different cell types a specific cell is still capable of becoming.
So how much potential it has left before it's completely locked in?
Exactly. The original fertilized egg, that zygote, and the handful of cells produced by its very first few divisions are what we call totipotent.
Toti meaning total.
Total potential, yes. A totipotent cell retains the remarkable ability to give rise to an entire, complete individual all on its own, including the placenta and supporting tissues needed for pregnancy.
Wait, really? Any single one of those early cells could just branch off and build a whole person?
They absolutely can, and we see a perfect natural experiment demonstrating this all the time. Identical twins.
Oh, of course.
When an embryo is still just a tiny cluster of these totipotent cells, if that cluster happens to split cleanly into 2 separate groups, each of those groups just carries on independently.
Because they both have total potential.
Because they are totipotent, they each have the power to develop into a complete individual.
And because they split from that exact same original cluster, they share the exact same original genetic recipe book, which is why they are identical.
Exactly, which is completely different from non-identical or fraternal twins.
Right, those are different.
Those occur when 2 separate egg cells are fertilized by 2 separate sperm cells at the exact same time. They are genetically no more similar than any standard pair of siblings, they just happen to share a womb. Identical twins are the direct result of totipotency in action.
Okay, so that a total potential is at the very, very beginning.
Mm-hmm.
But as the cells start reading those chemical GPS signals and, cascading their genes, I assume they lose that total flexibility.
They step down a level. As development progresses, they lose totipotency and become pluripotent. You have likely heard of these. They are the famous embryonic stem cells.
Right, stem cells. Pluripotent meaning plural, many potentials.
Exactly. They have many potentials, but not total potential.
They can still become a lot of things, but they can't build a whole human from scratch anymore.
Right. A pluripotent cell can still become essentially any cell type found inside the actual body, heart, liver, brain, bone, but it has lost the ability to build the supporting structures like the placenta.
Yeah.
From this point on, development is a one-way street of narrowing potential. The sources for our deep dive used a fantastic analogy here, describing it as a broad river dividing into progressively smaller channels.
Oh, I like that. You start with this huge, wide river of potential, and then it forks. And once a cell goes down one fork, say, the fork that leads to the nervous system, it can't swim back upstream to become a skin cell.
It just keeps branching into smaller and smaller streams.
Yeah.
The nervous system stream forks into brain cells or spinal cord cells. The brain cell stream forks into different specific types of neurons. It funnels down step by step until it reaches a very specific destination and becomes fully specialized.
So once the river reaches the ocean, once a cell is fully specialized and you're born, is the development story totally over, like the book is closed?
You would naturally assume the process stops at birth, but a limited version of this developmental engine keeps running for your entire life. Adult tissues retain small reservoirs of what are called multipotent stem cells.
Multipotent. So they have multiple potentials,
Yeah.
but it's highly restricted compared to the earlier stages.
They are far more restricted. They can only produce a limited range of related cell types, but they are absolutely crucial for your survival.
How so?
Well, your skin, for example, is constantly shedding, so it is being continuously renewed from multipotent stem cells deep in its lower layers.
Oh, right.
Your red blood cells only live for a matter of weeks. They are continuously replenished by multipotent stem cells hiding in your bone marrow.
So we basically have these little developmental toolkits left on standby all over our bodies, just quietly doing maintenance and repair, using the exact same logic of cell division and differentiation.
The underlying logic never really stops, but this
Yeah.
brings us to a crucial mechanical question. We've talked deeply about the chemical signals, the recipes, the GPS coordinates. We have a chemical map telling a cell it needs to be part of the spinal cord.
Right.
But knowing your coordinates doesn't magically build a spine. How do these static cells actually construct the physical three-dimensional architecture of a human?
Yeah, that's true. Because reading a chemical dial is great, but it doesn't explain how you get organs nested inside each other, or tubes, or a heart that actually pumps.
Right. The chemistry directly drives physical action. Morphogens don't just turn on genes that say be a nerve, they turn on genes that physically alter the cell's cytoskeleton.
That is skeleton,
Yeah,
like it's internal scaffolding.
exactly. This causes cells to actively migrate. Whole groups of cells detach from one location and migrate surprisingly long distances through the embryo to take up residence somewhere else entirely, helping to form the face or parts of the peripheral nervous system,
So the chemical map is literally telling them where to drive.
and how to change shape when they get there. The embryo doesn't just blow up like a balloon in place. Sheets of cells literally fold inward to form tubes.
Like origami?
Basically, yeah. Think about your entire nervous system. The spinal cord starts as a flat sheet of cells on the back of the embryo. The chemical signals tell the cells along the edges of that sheet to physically constrict, pulling inward like a drawstring on a bag. That flat sheet rolls up into a closed tube, which eventually becomes your spinal cord and brain.
That is staggering to visualize. I mean, this combination of chemical signals whispering instructions and cells physically migrating and folding themselves into origami shapes, it honestly sounds incredibly fragile.
It does sound like a house of cards.
Yeah. Given how many moving parts there are, cells dividing, migrating, folding, reading chemical dials, and over months and months across trillions of cells, you'd think the slightest hiccup would ruin the whole thing.
One of the most striking features of early development is how robust it actually is. Embryos have a staggering capacity to buffer against small disturbances.
Really?
Yeah. If a signal is a little too weak, or cell division is slightly delayed, or a few cells are lost along the way, the embryo doesn't just fail. It can usually compensate, course correct, and still produce a normally formed individual.
It's like it has built-in shock absorbers.
Exactly. Evolution has refined these gene networks over immense spans of time. Embryos that could tolerate a bit of chemical noise or minor mishaps survived better than those that couldn't.
That makes sense.
This creates redundancy. If one gene cascade is slightly muted, another overlapping pathway can often pick up the slack.
That robustness makes a lot of sense from an evolutionary standpoint, but it makes me think about everyday pregnancy advice.
How do you mean?
If the embryo is so incredibly good at correcting its own course, why are our doctors so exceptionally strict about what a pregnant person eats or what medications they take, especially in the first trimester?
Because that robustness has very hard limits, and it all comes down to a concept called a critical window.
A critical window.
A critical window is a very specific stretch of time when a particular organ or structure is actively being formed. When that flat sheet of cells is actively rolling up into the spinal cord, or when the cells are actively migrating to form the chambers of the heart, those tissues are highly sensitive to disruption.
So it's all about the timing of the construction schedule.
Timing is everything. A substance that disrupts development, whether it's a specific medication, alcohol, or a virus, is called a teratogen.
A teratogen, okay.
A teratogen usually only does its severe damage if the exposure coincides exactly with the critical window for the structure it affects.
So it's a timing issue.
Right. If the teratogen sweeps through before the heart starts forming or after it's mostly finished, the disturbance might be buffered by that redundancy we talked about. But if it hits right when those delicate morphogen gradients are trying to tell the heart cells how to fold, the error cascades.
Wow. So standard medical advice isn't just doctors being arbitrarily cautious. It is directly tied to the precise week-by-week schedule of when the body's structures are actively pulling themselves together.
It's grounded entirely in our understanding of that building schedule and how easily a specific step can be knocked off course if you introduce the wrong chemical interference at the exact wrong moment.
Looking at how complex this all is,
Yeah.
where does the science even go from here? Like, do we have this whole cookbook fully translated? Do we know every gene in every fold?
Yeah. Oh, not even close. This is very much an active, unsettled frontier of science. We still don't fully understand how a cell integrates all these countless conflicting signals, the morphogen gradients, the neighbor whispering, the physical tension, to make one clean, final decision about its fate.
It's just so much information for a tiny cell to process.
It is. We're also intensely studying how much of this is a rigid, hardwired instruction set versus cells just flexibly organizing themselves on the fly based on local conditions.
But how do you even study that? You can't exactly go in and tinker with the morphogens in a developing human embryo to see what happens.
No, absolutely not. But one of the most exciting recent developments is using pluripotent stem cells to grow highly controlled structures in the laboratory.
Oh.
Yeah, we can coax these stem cells to mimic aspects of early embryonic organization in a dish. It gives scientists a new, ethically considered way to directly watch, study, and even manipulate these earliest steps of life without needing actual embryos.
That's amazing.
But any confident claim that we have the whole picture fully worked out should be treated with deep skepticism. The field is moving incredibly fast.
Stepping back from all the specific genes, the morphogens, and the folding sheets of cells, what I find most awe-inspiring is that none of this requires magic.
No, it doesn't.
It is just ordinary chemistry and ordinary physics. Cells dividing, chemical signals fading out in water, genes switching on and off like light switches, cells physically pulling on each other. It is all totally normal biological principles, just repeated with such staggering coordination that it manages to build you.
It is the ultimate testament to the power of iteration. Simple biological rules applied over and over and over again, eventually creating unfathomable, beautiful complexity.
I love that. And I want to leave you with a final thought today, something for you to mull over that builds on what we discussed about those adult multipotent stem cells, the ones quietly doing maintenance in your skin, your blood, your gut lining.
They're always at work.
Because those cells are constantly replenishing your tissues based on that original DNA recipe, the physical matter that makes up you today is not the exact same physical matter you were born with.
That's very true.
Thanks to the ongoing, quiet engine of development running in the background of your life, your body is effectively a biological ship of Theseus. You are constantly rebuilding yourself from the inside out, cell by cell, until the very day you die. So the next time you curl your fingers into a fist or take a deep breath, remember that the awe-inspiring process that built you isn't just history, it's still happening right now.